right, everybody, surprise, the next time you guys come here on Thursday, we're going to have a test, all right? So I don't want it to sneak up on anybody, but next Thursday, this room's going to be pretty crowded, so I would suggest you get here early and pick out a nice, comfortable seat. What's going to happen is that we'll get things underway pretty much right on, spot on at 9:30. We'll hand out the tests and the answer forms that you'll get to fill out. And then you'll have the whole class to take the test. I'm anticipating that 90% of you will be done within an hour. And I know from past experience there will be 3 people who stick around till the very last minute. I'm not sure what they'll be doing, but we'll be here the whole time. Nothing's going to happen on Thursday except for you guys taking that first test. So there's no lecture, no party, nothing like that. Just come in here, take the test. You'll need a number 2 pencil to be able to fill out the Scantron form, and we'll get those tests graded for you within a couple of hours, and your grades will be posted on the pilot page for the course. You'll be able to see how you did pretty quickly. An answer key will be posted on the pilot page for the course. If you've looked at the old tests and you read the fine print at the beginning, you'll see there's always something about you can take this test home with you. We're going to give you a hard copy of the test. That's yours to keep, right? And what I would suggest, just, if it was me, what I would do is at the same time that you're writing answers down on the Scantron form, circle what you think was the correct choice on the hard copy of the test. Take that home with you, and then compare your answers to the ones on the answer key. And first, let me know if you think I've made a mistake on the answer key. And then second, once we've sorted that out, figure out if you didn't have the same choice that I did, why it is that you chose something different than what I chose. Right? But you'll be able to grade your own test pretty quickly and easily if you do that. 45 questions. No great surprise. It's going to look a lot like the tests you'll see on the pilot page for the course. I can tell you I've got it sent off to get duplicated right now, and it is 10 pages long. You'll get it as 5 double-sided pages, so we aren't even going to waste too many trees to be able to hand out these hundreds of tests on Thursday. Okay? For those of you who are taking the online version of the course, you're registered for Section 90. We have a special room just for you because the 120 Med Sciences room for the rest of the class can't handle that number of students. That's 101 Fawcett Hall, right? Nothing's changed there. So if you're in Section 90, you should go to that room to take the test. If you're not in Section 90, if you're here for the regular old-fashioned version of the course, 120 Med Sciences is the place to be. Any questions? Cool. My plan for today is to finish talking with you about phospholipid bilayers, membrane structures and functions. And as I said in class on Thursday of last week, I'm anticipating that I can do that pretty quickly, and then we can set aside some time at the end of class today as sort of a review session. I've got a couple slides that I've tacked on to the end of today's PowerPoint presentation just to kind of prime the pump and get me giving you the highlights of what I've talked with you about so far in the class. If you guys have questions during that sort of review session, I'm more than happy to take them, and let's get you guys in as good a shape as possible for that test on Thursday. If you got questions and you want to email me, I think you'll be surprised how quickly you'll get a response from me. If you really want to see me in person, I'll be around all day today. You shouldn't have too much trouble doing that. I think I'm going to be in Columbus on Wednesday tomorrow, so you may have a hard time finding me, but email will work. Okay? All right, so we'll get to work. There is one other thing, though, that I wanted to just mention to you. I understand that a week from Thursday, so I think that's October 6th, there are going to be some camera crews that are going to be in here in this lecture hall. It's for a obscure cable TV show. All right, Michelle Malkin investigates. I think she's a friend of Sean Hannity's or something like that, as best as I can tell. But If you want to be on an obscure cable TV show and tell your parents that, look, I have been coming to class, look nice on Thursday of next week. All right, show up and look nice on Thursday of next week. If you'd rather not be on an obscure cable TV show, stay home on Thursday of next week, or sit real far in the back and I'll dim the lights so you can hide back there. Okay? Hey, speaking of which, I'm glad that I was thinking about that. I promised the producers of the show, so here, this is your one chance at being famous right now. How about that? There, they wanted to see what the room looked like. My kids are really good. Here we go. There, now we're talking. All right, that's going on the internet for sure. All right. So, okay, so that's Thursday of next week. The people who are listening to the Integrity recording have got to be wondering what's been happening for the last 10 seconds. So that's okay. You guys get that bonus secret knowledge. Okay, so Thursday of next week, if you want to have a brush with fame, look pretty, and you'll get to be on this cable TV show. Okay, but here's the real business that we got to worry about here today. I want to just wrap things up, and that's not such a bad phrase to use to start this lecture, by talking with you about membrane structures and functions. And that's going to set us up then so that when we get back to talking about biology things on Tuesday of next week, we can start then talking about what makes living things tick, how it is that they control their internal environment and get to do things that non-living things have a hard time pulling off. But today, again, let's wrap things up, literally in some sense, by talking about membranes. Plasma membranes, again, are this boundary that defines where the cell, a living thing, begins, depending on what your orientation is, and where it ends, right? On the outside of the plasma membrane, it's a diffuse, chaotic sort of world. On the inside of the plasma membrane for a living cell, you've got a higher concentration of things that living things value. You've got proteins that are catalyzing reactions. The world's different on the inside of the plasma membrane as opposed to the outside. That plasma membrane is a boundary. And it's also one of the most obvious things that you see when you use a light microscope to take a look at a cell. So it shouldn't surprise you to hear that from pretty much the very beginning of microscopic analyses of cells, people were kind of interested in what's going on with these plasma membranes. And as long ago as 1895, some guy named, well, here, we'll get to that in a second, some guy named, let me make sure I get his name right here, Charles Overton. The fact that I had to look at my notes means it's not stuck in my head, right? That means it's not going to be on the test. That's that level of detail. But a guy named Charles Overton in 1895 had this sort of interesting idea. He did some experiments, and he came up and he put forward this idea that plasma membranes, cell membranes, were likely to be composed primarily of lipids. And he arrived at that conclusion from a fairly simple set of experiments. He figured out that if you had molecules, specifically dyes, that were fat soluble, in other words, were hydrophobic, that they got to the inside of cells faster than molecules, dyes, that were hydrophilic, that were water soluble. So if you had two different dyes, let's say you set this up. You guys have never done this, I'm sure. You'll have a chance to do this later this semester. We're going to have you doing gel electrophoresis in lab. And one of the gels that you're going to electrophorese is literally Jell-O, right? And one of my favorite Jell-Os to electrophorese is lime Jell-O, because it turns out that lime Jell-O has 3 different dyes in it. And they have different charges and different molecular weights. So 2 of them move in one direction, 1 moves in the other direction when you put an electric current across it, and you get this neat separation. How about this? This is essentially Charles Overton's experiment. He took 2 different dyes, each with a different color, one of which was fat soluble, one of which was water soluble. He mixed them together as best as he could. He put them into contact with living cells, And he watched what happened. The living cells pick up the fat-soluble dye in its color much faster than they pick up the water-soluble dye and its color. Right? And so he's looking at this, he's thinking, how can this be? And he comes to this interesting conclusion that, hey, the reason the fat-soluble stuff gets in faster is because it's moving through something that's fatty itself. He comes up and he puts forward this idea that plasma membranes must be predominantly lipids, fats, because that's why it is you get that ease of transport of fat-soluble things. The fat-soluble stuff can move through it. The water-soluble stuff has a hard time. So there's his clever idea in the late 1800s. About 20 years later, some other folks came along. They're working with Charles Overton's ideas. They come along and they do some actual experiments. They get some plasma membranes and they look to see just what stuff are we holding when we get plasma membranes. And literally, they were holding some plasma membranes back then in the early 1900s. This isn't lots of, gas chromatography or anything like that. They're literally working with chunks of stuff to figure out what's going on at this fine scale. May gross some of you out, but the membranes that they were working with were red blood cell membranes. And this is actually the basis for an awful lot of experiments. We know an awful lot about hemoglobin because hemoglobin is easy to isolate when you go to a slaughterhouse and you can get barrels full of blood. That's what these guys did in the early 1900s. They got a lot of red blood cell membranes simply by going to a slaughterhouse, getting buckets, barrels full of blood, throwing away the water and asking what's left, right? What's the membranes that are left at the end of this process? Red blood cell membranes. And you know what they found? They found Charles Overton was right. Good job, Charles Overton. They found that plasma membranes were made. There was a lot of lipids left behind. But they found one other thing, too. They found that there was also a suspiciously large amount of protein. In fact, by some estimations, they found there was as much protein as there was lipid. Usually about half as much protein as lipid, but sometimes when they did these experiments, they found the plasma membranes were largely lipid and still quite a bit of protein. Right? So a protein-lipid mix. And that insight sets the stage for another pair of guys to come along, Davison and Danieli, and they create this model conceptually of what a plasma membrane must look like. So let's see, I got some pictures for you here. That's what a plasma membrane looks like by today's reckoning. We'll see more and more of this. That's going to be sort of like the last slide you see in this PowerPoint presentation as well. But here is a phospholipid bilayer. So Charles Overton's actually the guy who first starts talking about these must be lipid-based membranes. Check out what's going on, right? You got your phosphate group up here at the top. 2 negatively charged oxygens should be hugely water-soluble. You've got a chain of hydrophobic fat that dangles down, 2 chains from each phosphate group, and you get this water exclusion zone in the middle of the plasma membrane, a phospholipid bilayer because there's 2. And then on the other side, you've got more phosphates for the other side of the bilayer and a watery environment over there. I'll tell you what, I've been arming you guys with some cool sounding words. My favorite by far is the reticuloendothelial system, right? It's hard to top that one. I've got something coming up. Okay, I'll tip my hand. In another month, I'm going to introduce you to a synaptonemal complex. That's another good one. But for today, let's focus on reticuloendothelial system. And here's one other. I don't know that this is in the same league, but it's still a pretty cool word, amphipathic, right? Amphipathic. What that means is it's a word that you can use to describe a phospholipid. Phospholipids are like the paradigm. It is the stereotype. It is the perfect example of a molecule that's amphipathic. In a little bit less formal terms, you could say phospholipids are schizophrenic. Right? They have this dual personality. They've got this intensely water-soluble phosphate group at one end, and then they've got this intensely hydrophobic pair of tails that dangle down from them. Right? That's what we mean by amphipathic, a molecule that has one part that is very water-soluble, behaves that way, and we have another part that's very fat-soluble and hydrophobic, behaves that way. So we got these phospholipid bilayers made from these amphipathic phospholipids. Right? You got one layer here, you got another layer there. And Davison and Danieli come along, they build on Overton's observation that there's a lot of lipids there. They incorporate these new insights in the 19-teens, I suppose is the way to describe that. It's like around 1915. They get this new insight that there's also proteins that are involved. And Davison and Danieli take these two pieces of information and they come up with the Davison-Danieli model of plasma membranes that is what is shown on the screen right now. Phospholipid bilayer in the middle, and then on top and below, a layer of proteins. The Davison-Danieli model is wrong, okay? But it held sway for about 50 years. For about 50 years, people thinking about what plasma membranes looked like would always trot out pictures that looked like this. Essentially, a phospholipid bilayer sandwich where the phospholipids were the meat and the proteins were the bread on either side of the sandwich. All right? It's cobbled together because of those two observations. Plasma membranes must have some fats associated with them because fat-soluble dyes move through more quickly than water-soluble dyes. And when you look at red blood cell membranes, you find there's a lot of protein there as well as a lot of phospholipid. You put those two sorts of ideas together and you get the Davison-Danieli model of a sandwich. But I've told you the Davison-Danieli model we now know is wrong. And how we know that it's wrong is because in the 70s, for the first time, microscopes got to the point where you could actually start to look at membranes at very fine scale, at a small level of detail. And I'm talking now specifically about the advent, the origin, the availability of electron microscopes. And so for the first time, people are able to really look carefully at the structure of a plasma membrane. And the first glimpses told them something kind of surprising. Those first glimpses with electron microscopes revealed that plasma membranes were about 50 atoms thick. All right? So that's the sort of scale that they were able to look at, 50 atoms thick. And it turns out that this Davison-Danieli model is way more than 50 atoms thick. You got your oxygen here for the phosphate group. You got a phosphorus. You got another oxygen, so that's 3 atoms. You've got a chain of about 15 to 20 carbons here, right? So you've got, what, 25 or so atoms for this phospholipid. You get another 25 or so carbons for this phospholipid. You put those two together, you're at 50, right? And yet, nowhere have we accounted for what's going on with the proteins. The scanning electron microscope images of phospholipid, of plasma membranes from things like red blood cells showed that the thickness was determined just by the phospholipid bilayer itself, and the proteins just didn't add up, right? Because the proteins are going to be dozens, probably scores of additional atoms thick. This model had to be wrong. So now, in the 70s, another couple of guys, Singer and Nicholson, come along, and they've got 3 pieces of information. They've got Overton's idea that the plasma membrane must have fats in it. They've got the insight from red blood cells in the early 1900s that there are proteins that are involved. And now they have this third piece of the puzzle that tells them that the membranes are actually only 50 atoms thick, only thick enough really to be a phospholipid bilayer. What are you going to do with the proteins? You know there are proteins involved. Where are the proteins? Where are they hanging out? So Singer and Nicholson come up with what they now call the fluid mosaic model. That's what they do with the proteins. Instead of having the proteins like Davison and Daniele did as sort of the pieces of bread on the sandwich, they get the proteins embedded in the membrane itself. 50 atoms thick, but now, and it meets the rest of the criteria, 50 atoms thick, mostly phospholipid, but still there are a lot of proteins there. Where are the proteins? They're bobbing around inside of the phospholipid bilayer. This was actually kind of a radical proposal when it was made in the 70s by Singer and Nicholson. And what they were running up against was this stereotype, this erroneous understanding that proteins themselves were water-soluble. All the proteins that people had been studying for years up till that point had been the ones that were the low-hanging fruit, the ones that were easy to get a lot of, to take a look at, to analyze, to beat up, to characterize, right? Proteins like hemoglobin. You can get buckets of hemoglobin, and hemoglobin had been well studied. Hemoglobin is very water-soluble. Other proteins that biochemists had been studying were also very water-soluble. And it hadn't really occurred to people that there may be other kinds of proteins that were maybe harder to isolate, but behaved differently. And maybe they behaved differently because they were harder to isolate, the ones that are more fat-soluble than water-soluble. And so Singer and Nicholson put forward this model, the fluid mosaic model, and they sort of conjecture, they speculate, maybe there's a bunch of proteins out there in the world that we haven't been paying much attention to that actually are not very water-soluble, that are more fat-soluble than water-soluble. And they throw this idea out there, and sure enough, people start to look more carefully for these kind of proteins, and sure enough, they exist. And in fact, most of the proteins that are associated with red blood cell membranes, as well as other membranes, are these kind of proteins that have big patches on their surface that are hydrophobic, so that they can nestle up against these fatty lipid acyl groups for the phospholipid, the lipid part of the phospholipid bilayer, and then other patches that have more hydrophilic, even charged amino acids, so that they can hang out in the watery environment outside of a cell or inside of a cell. So we now understand that an awful lot of proteins associated with membranes are just like these, where they span the whole membrane. Some of them hang out just in the lipidy part. Some of them hang out just on the phosphate part. But most of them are these proteins that themselves fold up into shapes that cause them to present on their surface combinations, collections, assemblages of amino acids that make them really comfortable hanging out in the lipidy part, the fatty part of a phospholipid bilayer. So, this is a moot question in light of what I've just told you, but I think it's a fair question. It's the kind of question I'm going to ask you guys. In fact, I may have asked a question very much like this on the test that you're going to see on Thursday. But let's kick this around a little bit, see what you think. So I give you a couple of different kinds of amino acids, and I ask you which ones are most likely to be present in the transmembrane domain of an integral membrane protein. There's some jargon here, right? What is a transmembrane domain, and what's an integral membrane protein? Let's just back this up. This is an integral membrane protein. That's an integral membrane protein. That's an integral membrane protein. It's integral in the sense that it is physically part of that membrane. That's all that integral here means. It's part of the membrane. And transmembrane domain, you've heard of the Trans-Siberian Orchestra, right? Trans-Siberian goes across all of Siberia, right? Well, a transmembrane domain, the protein, this domain spans the entire width of that phospholipid bilayer. It's a transmembrane domain. So Shouldn't be too much mystery as to what transmembrane domain means. Shouldn't be too much mystery as to what an integral membrane protein is. What kind of amino acids do you expect will be on the surface of this protein in this part of the protein, the part that spans the membrane? Super easy, right? Insultingly easy, perhaps, at this point in the class. You got 4 kinds of amino acids that we've talked about, right? I've said the categories are where the action is for the purposes of a class like this. You've got your nonpolar amino acids. You've got your polar amino acids. You've got the positively charged, and you got the negatively charged amino acids. Let's work backwards. Negatively charged amino acids, are you crazy? Could there be negatively charged amino acids there? Those lipids would freak out if there was something with a negative charge there. They would form an exclusion zone around it. They'd push it away. They'd try to nestle up against other fatty things. Negatively charged amino acids just couldn't handle it there. They would be shunned at a molecular level. Nobody wants to be shunned, right? Not a good choice. Ditto for the positively charged amino acids. Lipids want nothing to do with those. They will exclude them. They'll push them aside and nestle up instead against other fatty things. Polar amino acids. Well, it's not so heinous a shunning as what would happen for a positively or negatively charged amino acid. But polar, that's just not what lipids are about. What are we left with? The nonpolar amino acids, that's match made in heaven, right? Nonpolar means that they also are afraid, they're hydrophobic, they're afraid of water, they don't want to have anything to do with water, they like to hang out with other hydrophobic things. What other hydrophobic things are there? Oh my gosh, lipids, like those in a phospholipid bilayer, right? So nonpolar sounds like a really good choice for the amino acids that you would expect to find on the transmembrane domain of an integral membrane protein. Is that a choice? Charged amino acids like glycine? No. Polar amino acids? Probably not. Special amino acids? I wonder what makes them special, like glycine or proline. Hydrophobic amino acids like valine, that looks like a winner. Any of the above with no preference, that looks like somebody wasn't being very creative and coming up with choices to me. So of the choices I've got, this one sure looks like a nice one. That's what I would go to as the right answer for this question. What if I changed the question? What if instead of talking about the transmembrane domain, I focused your attention on the external domain, the part of the protein that pokes above the phosphates in the phospholipid bilayer. Different answer, right? One word change, I get a whole different answer. These should be those hydrophilic amino acids up in here, the charged ones, the ones that are polar. Non-polar amino acids just wouldn't be very comfortable being forced to poke out into the watery environment up here. Also not very comfortable poking into the watery environment here. You want to make it really comfortable? How about if you dress up this part of the protein with some positively charged amino acids so that they can hang out with the negatively charged phosphates of the phospholipid bilayer? Man, now that's a happy relationship. So there you go. That's the Davison-Danieli model, it's called. They learned a lesson from Singer and Nicholson because Singer and Nicholson were derided for quite a while, fairly mercilessly. They had built their careers for decades around the Singer and Nicholson model, the Davison and Danieli model that had the sandwich going on, right? Davison and Danieli were made men. They were in good shape for their whole careers. And then these upstarts, Singer and Nicholson, come along and they say, you guys have been wrong for decades. This is the way membranes really are. They learned a lesson. Davison and Danieli learned a lesson from what they did to Singer and Nicholson. They don't call this the Davison and Danieli model. They call it the fluid mosaic model, so that someday if they're proven wrong, it won't be their names that get dragged through the mud. It'll just be the name of this model. But the model's name is a pretty good description of what's going on here. So it's a fluid mosaic model. And let's talk about that fluidity for a few minutes now. Here again is a phospholipid bilayer. Phosphates at the top, lipids in the middle, phosphates at the bottom. It is super easy for one of these phospholipids to switch spots with another phospholipid laterally. What's to stop this molecule, this phospholipid, from changing places with this phospholipid? It's just a matter of, you want to be over here, that's fine, I'm going to step aside, you move in there, and I'm going to take up your spot. No problem with that kind of lateral movement. No resistance to that at all. And that is in striking contrast to this kind of swapping of phospholipids. To do a flip-flop, like is shown in this part of the slide, what you got to do is you got to somehow or other persuade this phosphate group on this amphipathic molecule with radically different behavior and affinities from one end to the other. You got to somehow persuade this negatively charged, intensely negatively charged phosphate group to work its way through the profoundly hydrophobic interior of the membrane. How are you going to pull that off? And then through all the way to the other side, and at the same time, pull the same trick on this phosphate group and get it to work its way all the way through the hydrophobic space there until it gets back to the hydrophilic space. It turns out that is crazy difficult to do. The energy that needs to be expended, it's so counter-natural. Nature doesn't want these hydrophobic things brushing up against the hydrophilic things. It just can't work often at all. So lateral movements of these phosphates and the phospholipids, super easy. Flip-flopping movements just isn't going to happen in any sort of realistic time scale. And so what about the proteins? What are they doing in this sea of phospholipids? Well, here's another picture of a phospholipid. This is probably an even better picture of a phospholipid than the one I was working with before. Check it out. These transmembrane domains almost always have alpha helices that define them. So we talked about alpha helices and beta sheets. Check out all those pretty alpha helices there. Can't get too much prettier than that. Notice how the length of the alpha helix, the amino acids coil up in just the right way to span the membrane, not that much more. Notice what kind of amino acids should be on the surface of these coils. It should be the hydrophobic guys. Maybe a positively charged one or two over here, maybe a positively charged one or two over here to get a nice anchor with the negatively charged phosphate groups. And then a bunch of polar amino acids up here, maybe some charged ones, a bunch of polar ones down here, maybe some charged ones. How's this protein going to behave? What's it going to do? What do you think? What if we tried to flip-flop this protein? Notice the N-terminus, that's the beginning of the protein. That's where the protein, that's the first amino acid that got linked together when you made this long chain of protein. The last amino acids, that one down over here, poking down at the bottom. What are the prospects of taking this protein that must have been synthesized, by the way, by a membrane-bound ribosome in the rough endoplasmic reticulum, harkening back to things I've talked about the last couple of lectures. What are the prospects of taking this protein and flipping it, right? Getting the amino terminus to the inside and the carboxy terminus to the outside of the cell. This is the way it got made. The membrane-bound ribosome in the rough endoplasmic reticulum started here, then made something that spanned the membrane in this direction, then made something that spanned the membrane in this direction, then made something that spanned in this direction, this direction, this direction, this direction, this direction, somehow or other wraps around down here to the carboxy terminus. And that's when the ribosome stopped making it. What are the chances that you would take this protein and flip it? Get the nitrogen, the N-terminus flipped around to the inside of the cell and the carboxy terminus to the outside of the cell. Virtually no chance at all. For the same reasons that flip-flopping of the phospholipids can't happen, you can't have the orientation of this protein change either. The ribosome that translated it gave it a particular orientation. That's the way it's going to stay for as long as that protein stays a protein. Someday it'll get broken down by a lysosome into its component amino acids, but until that happens, that's the way it's set up. And you won't get any of that flipping around. And because of the way that ribosomes do this synthesis, every time they make a copy of this protein, so there's not just one of these proteins in the membrane, there's probably hundreds, thousands. All of the proteins of this kind will have been made the same way. The ribosomes will have always started here. They'll have made these. These will have naturally passed through the membrane because they're hydrophobic. They don't want to hang around out there, so they go through the membrane. The next ones that get made also want to be in the membrane, but there's a little stretch that's hydrophilic, so they poke out over here. Every time a ribosome makes a protein like this, it's going to make it the same way. That orientation is always going to be the same for every copy of that protein that gets made. And they'll stay the same for, again, the life of the protein itself. So flip-flopping, not going to happen. Lateral movement, sure, why not? Is there anything more appealing about this particular spot for the protein as opposed to this spot? You could scoot it over to the left. You can scoot it to the right. No problem. Flip-flopping, not going to happen. So the question is, do membrane proteins move? Folks set up an interesting experiment. They took mouse cells and they took human cells. Would you be surprised to hear that the proteins on the surfaces of mouse cells are recognizably different than the proteins on the surfaces of human cells? Nah, of course. That's part of what makes us different from mice, is we have different proteins on the surface of our proteins. They took these two different kinds of cells, they smushed them together to make one big cell, and they asked, well, what happens? Do all the mouse proteins stay on one side of the smushed thing and all the human proteins stay on the other side? What do you guys think is going to happen when you do this? Flip-flopping, no. Lateral moving around, yeah, why not? So check out what happens. You smush them together, you make this hybrid cell. They didn't use the nasty word here. That's a chimera, all right? It's sort of like a half mouse, half human cell now. But you smush them together to make that hybrid chimera. And within an hour, when you look at these cells, there's an even distribution of human and mouse proteins. Why? Because lateral movement is easy. Flip-flopping, just not going to happen. So, it's a fluid mosaic model. When we're talking about fluidity, we're talking about the ease with which things move from left to right. In those two dimensions, lateral movement is super easy. It's a mosaic because when you look down at it from above, you see this patchwork, right? You guys know what mosaics are, right? That's what we're talking about here. You take little pieces of tile of different colors and you put them together in a way to make a picture so that when you look at them from a little bit of a distance, like, ooh, pretty picture, right? You know, that's what's happening here. It's fluid. The pieces of the tile can move around, but it's a mosaic because when there are little pieces bobbing around in this phospholipid bilayer. So, we've talked about a bunch of different kinds of proteins. When you put it all together, what we end up seeing for animal cells, plant cells are similar but a little bit different. When you put it all together, this is what our current and probably the correct understanding of a phospholipid bilayer is, of a plasma membrane is. Check it out. The most prominent feature, perhaps, is the phospholipid bilayer itself. One phospholipid here, another phospholipid layer here, bilayer, two layers, a phospholipid bilayer. But along the way, there are proteins, integral membrane proteins that bob around in this sea of lipids, the plasma membrane. And there's some that don't make it all the way through to the other side. There are some that are called peripheral proteins that really don't interact with the hydrophobic part at all. They're just touching other proteins or the phosphate groups, but they're still associated with the membrane. A lot of the proteins that are integral proteins they get dressed up with sugars attached to them. You might recall when I talked with you about the Golgi apparatus, I mentioned at one point that protein processing occurs there. Among the processing steps that happen is the attachment of sugars to one side, to one of the exterior surfaces of the protein. The part that pokes out into the world often gets glycosylated, meaning sugars get strung on to the outside of it. Not a ridiculous number of sugars, usually 15 or less, so a relatively small number, but nonetheless, sugars get attached to a lot of these integral membrane proteins on the part in particular that faces out from the cell. What do those sugars do? Well, very simplistically, they make the surface of the protein they make the surface of the protein and the cell sticky. Right? So those sugars make this sticky glycohelix that helps one cell stick to another cell. Not a bad thing to do if you want to be a multicellular organism. But they also have some functional characteristics associated with them. Let me tell you what I know, and then I'll tell you what I don't know. What I know is that for any given integral membrane protein, for that particular kind of protein, the sugars that get attached are probably always the same. What I don't know is how cells know, how the Golgi apparatus in particular knows what sequence of sugars to put onto the surface of a protein. That's an open question for biology. A professor that I knew in graduate school studied this. He's been studying it for like 30, 40 years now. He assures me that gaining the insight in terms of how it is that those sugars get attached and how it is that cells know which sequence to put on, figuring that out is going to crack open as much stuff as being able to figure out what the nucleotide sequence of genes is. All right? I don't know if he's right about that or not, but I can tell you, despite lots of work, people still haven't figured out how those sugars exactly get strung on in a particular way. But I can tell you that most of the sugars for any given protein, from one copy of that protein to another, will have the same patterns of sugars associated with them. And I can tell you that aside from just making the surface of the cells sticky, they do have some functional importance beyond that. I'll bet most people in this room know your blood type in terms of ABO. You know what that boils down to? It boils down to a difference in the sugars that you attach to one of your integral membrane proteins for your red blood cells. If you are blood type A, that means that your proteins, that particular protein, are displaying a particular combination of sugars that we call type A. If you're blood type B, that means that you're displaying a particular combination of sugars we call type B. If you are type O, that means you don't have any sugars on those particular proteins. And if you're type AB, that means that both the A kind and the B kind are out there. Right? And so there's an immunofunctional role for those sugars, but you can imagine how those sugars might play some interesting catalytic roles or functional roles on top of that. And then there are some fibers of the extracellular matrix that also help interact with these sugars and tie everything down. But you can see the phospholipid bilayer, the fluid mosaic model, gets all dressed up, and there's a lot of stuff going on there. There's a lot of interesting questions that still need to be answered, and this is still a pretty active area of research. One more time, here's a picture of the Golgi apparatus. This is where it is. The sugars are getting attached, and the proteins are getting glycosylated. So, peripheral glycoproteins is what I've just been talking with you about. Let's talk about some of the functionality then for these membranes. Here is the fluid mosaic model again, sort of a simple look at it one more time. Let's just get back to basics and even more basic and talk about what's going on with the membrane. In lab, all last week, you guys played with semi-permeable membranes. All right? The deal is pretty much this. If you've got a membrane that sets up a barrier from one side of a tube or a beaker to the other side of the tube or beaker, you can have a membrane that is completely permeable and allows the molecules to move freely from one side to the other. Shouldn't be a great surprise to anybody that if you start out with a high concentration on one side and a low concentration on the other side, that over time, diffusion will take place and you'll get an equal concentration of the molecules on both sides of the membrane. It might help to, that's intuitive. We've known since kindergarten that diffusion happens. If you take a drop of dye and you put it into a glass of water, you can see the drop of dye at the beginning. After a few minutes, it gets more diffuse. After a few hours, the whole cup is whatever the color of the dye was. Intuitive, simple. I want you to think a bit about what's happening at a molecular level. Why is it that diffusion is taking place? At the start, check it out. There's more of these little balls on this side of the dividing line, the membrane, than on this side of the membrane. It's easy for a ball to move from this side to that side, right? It's a one-way street. There are no balls over here to move over to there, so you get a net movement of molecules from this side to this side. But eventually, when you get the same number of balls on both sides of the membrane, for every one that goes this way, there will be one that goes that way. And that's when you've gotten an equilibrium, when there's an equal amount of movement from one direction to the other. And you can do that in two directions, right? You could start out with, what do we want to call these? Yellow balls on the left, purple balls on the right. If you have more purple balls on one side than the other, there's going to be a natural tendency for us to see net movement of purple balls to this side and yellow balls to this side. And sure enough, you'll end up with that kind of equilibrium. Let me just quiz you one more time, getting you to conditioning you for this test on Thursday. Which of those kinds of molecules do you think is going to work its way across a fluid mosaic monomembrane faster? Water, oxygen, phosphates, glucose, or sodium? I can tell you, none of these look to me like they're going to be something that's going to have a particularly easy time. Well, actually, that's not true. Moving through a membrane. All right, I need to correct what I was just saying there. One of these molecules is different from the others. It's a hydrophobic kind of thing. It's involving only nonpolar bonds, right? That oxygen gas should have a pretty easy time working its way through the lipid part of the phospholipid bilayer. All right? But it's kind of interesting, right? Because on that list of molecules that I've got for you there, how important is it for a cell to get oxygen inside? Well, that's not necessarily a bad thing. But I'll tell you, if a cell had a choice, I could have either an oxygen gas molecule or a glucose molecule. Cells are going to want the glucose every time, right? Because the glucose is money, literally money in the bank for a cell. It's an energy-rich molecule. Glucose, it just doesn't look like it's going to be able to make its way across a membrane very easily because this is a pretty water-soluble molecule. So the question then starts to become, how do you get molecules that cells want and need, like glucose or sodium or phosphate groups or even water, from outside the cell to inside the cell? Given what you know about membrane structures, how's this going to work? So moving in that direction, check out this neat arrangement. This is a test tube that's bent into a U. Okay? There are such tubes. And you can put a membrane at the bottom of the U. that separates one side of the tube from the other side of the tube. And you can fill them both up with water, put a little bit of sugar in one side, a lot of sugar in the other side, and let's say that this membrane is semi-permeable, not permeable. Permeable would mean that both water and sugar, those are the only two players on this experiment. Permeable would mean water and sugar could both move through the pores of that membrane. But this is not permeable, it's semi-permeable. And what the rule of the game will be is this, is if the pores are small enough for water molecules to fit through, but not allow sugar molecules to fit through. Sugar molecules are bigger, they can't fight their way through the holes in the membrane. Water molecules are smaller, they can go freely from one side to the other. What's going to happen when you set up this experiment? Only two molecules in play. Green balls that are sugar, things that aren't balls that are water, right? The only two kinds of molecules. Water molecules can move through, sugar molecules cannot move through. At a molecular level, look what's happening at that membrane. Frankly, surprisingly perhaps, I think this is pretty exciting. But look what's going on here. There's water molecules on this side that can move through. There are water molecules on this side that can move through. But there are more water molecules that are free to move through on this side than there are water molecules on this side that are free to move through. A lot of the water molecules on this side are hanging out with, tied down with, sugar molecules that can't move through. There's more water molecules on this side of the membrane. There's a net movement of water molecules from this side over to that side at a molecular level. Do you see that? I mean, that's just what the picture's showing you. There's more water molecules that can move from left to right, that can move from right to left. At a molecular level, that's because the water molecules on the right, more of them are tied up, hanging out with sugars, and they can't break free of the sugar to move across the membrane. They're just tied up. There's more water molecules on one side than the other. What's going to happen? There should be a net movement of water from this side of the tube over to that side of the tube. Seriously? If there's a net movement of water from this side of the tube over to that side of the tube, what's that going to look like in the real world? I think what that's going to look like, and this is kind of surprising, there's almost a magic aspect to this. I think what that's going to look like is the water level over here is going to go down, because water's moving from here to here, and the water level on this side is going to go up, because water's moving from here to here. Seriously? I can get a higher column of water on this side of the tube than on this side of the tube just by putting some sugar into this side of the tube? From a molecular sort of theoretical way of thinking about it, the answer to that question is yes. And it turns out that when you actually do that experiment, that's exactly what happens. This seems counterintuitive to me. It's not the sort of thing I would have guessed would be the way this sort of thing. I didn't think the world worked like this. But it's because I guess I don't have a lot of practical experience working with semi-permeable membranes that can be these molecular sieves like we've been talking about here. But there's a net movement of water from one side of the tube to the other. There's a name for this phenomenon. It's called osmotic pressure. If you have a higher concentration of stuff on one side of a semi-permeable membrane than the other, water is going to osmotically move from one side of the membrane to the side where there's more solute, and it's going to raise that column of water. And it's directly proportional, the pressure head that you get, to how high you can drive that column of water, is directly proportional to the concentration of the solutes on both sides of that semi-permeable membrane. So I'll tell you, when I first encountered this idea of osmotic pressure, I remember it very well. I lost like 2 nights of sleep about this particular revelation to me when I was a freshman as an undergraduate. Right? It's like, no way, this can't be how the world works. All right, now I found a chink in the armor. I got this piece of knowledge, and I can use this in a way that's going to let me rule the world. Right? Check out what it is that I came up with for my way to dominate the world with this insight. I still think, frankly, in all modesty, I think this is hugely clever. And I want you to tell me why my clever idea doesn't work. What I want to do is this. I want to take this, it's called a YouTube. Before there was a YouTube, this is what we called these things. I want to take this YouTube, and I just want to modify it just a little bit. What I want to do is I'm going to take my YouTube, and I'm going to add another YouTube up here to the top, right? No membrane here, but can you picture what I'm talking about? I've got a U with another loop that comes over to the top. And I'm going to have the other end of the U end over here. No membrane here, just lots of sugar, let's say, in here, but a semi-permeable membrane at the other end of the tube. Huh? See what might happen here? If I get a high enough concentration of sugar over here, all right, and a low enough concentration of sugar over here, but a good supply of water, right? I may be able to get this osmotic pressure, the head, to push all the way around to the top of the tube to the next semi-permeable membrane, which will keep the sugar in this part of the tube, but allow the water to drip out on that side of the tube. Huh? Huh? You guys see what's happening now? And then all I got to do is set up a little, pinwheel down here. So as the water's flowing out the other end of the tube, the pure water's flowing out the other end of the tube, it's spinning around a little dynamo. I'm charging a battery. You know what I've just described to you? I've described to you a perpetual motion machine, a machine that runs and produces energy at no cost forever. You set up this tube one time, you walk away, and your dynamo is working there, and you're cranking out energy. Saudi Arabia, the Middle East, they're out of business. I'm rich. I can fly to the moon with this thing. It's amazing what you could do with such a machine. And it proves hundreds of years of people saying that there's no such thing as a perpetual motion machine wrong. There's got to be a Nobel Prize in that, right? Well, guess what? I set that up as an undergraduate, as a freshman. I went to a chemistry professor and I said, okay, I learned about osmotic pressure in your class. I want to check this out. Can I really get this? Oh, yeah, here's a membrane, here's some YouTubes. Use some tape, tie one YouTube to the next YouTube, set it up. And guess what? It worked for about a day, and then it stopped. It's like, what the what? Come on, keep going. Why is it stopping? What's happening here? And the chemistry professor says, well, I don't know why it's not going to work, but I know it won't work because you can't have a perpetual motion machine. That's the first law of thermodynamics. We'll talk about that on Tuesday. A week from today. And so it's like, but why is it not working? Do you guys have any ideas on why my perpetual motion machine stops after a day or so? I'm tempted to leave that just burning at you for a week so that when we come back on Tuesday after the first test, you'll be interested in figuring out the answer. I don't know, it could go either way here, but I'll tell you why it doesn't work. Ultimately, what ends up happening is on the other end of the flipped U-tube, there's a semi-permeable membrane, and water does come through, but what ends up happening is that because of friction between the water bumping into the membrane, it doesn't come gushing out. It forms like a little drop and drips out. But while it's hanging there before it drops out, there are water molecules at a molecular level that are at high concentration on one side of the membrane and low concentration at the other, and they fight their way back and they set up an equilibrium at the other end. And so you get this little drop of water on the one end, and it just sort of hangs there, because that drop has water going back and forth, and darn the first law of thermodynamics, right? But if not for that, man, I would have been I wouldn't be here today if that actually had worked the way that I had thought that it might for about 48 hours in my youth. So, enough about me, but let's talk now about some other practical implications of osmotic pressure. It's a big deal for living things. There are different concentrations of solutes on one side of plasma membranes relative to another side of a plasma membrane. Cells have to reconcile with that all the time. And I want you to remember, if nothing else, from my little perpetual motion story, there's pressure. There's osmotic pressure that's involved. And it works out to something like this. Here is a normal, happy-looking red blood cell. If the concentration of solutes on the outside of the cell is the same as the inside of the cell, you've got the same number of water molecules going into the cell across the plasma membrane as are going out. You're in equilibrium. We call that an isotonic solution. That's the way that you would hope it always is, right? But let's say you find yourself in a circumstance where you've got more solute outside of the cell than inside the cell. So there's a higher concentration of stuff outside than inside. There's going to be, oh wait, it's the other way around. Higher concentration inside the cell than outside the cell. What's going to happen is you're going to have a net movement of water into the cell, and that in turn will ultimately puff up the membrane until it blows up. This solution here is called a hypotonic solution. That means that there's a lower concentration of solute outside than inside. And the flip side of that coin is if you have a circumstance where there's a higher concentration of solute outside the cell than inside the cell, there's a net movement of water from outside to in, the cells shrivel up. This kind of solution is called a hypertonic solution. So the concentration of solutes across a plasma membrane is an important thing to keep in mind. They don't have to be the same solutes. It certainly makes it easier from a bookkeeping perspective if they are. But it's the number of free water molecules that's really the important thing if that's what the membrane is permeable to. If the membrane is permeable to the movement of water, it's the number of free water molecules on one side relative to the number of free water molecules on the other side. And so if the water molecules on one side are tied up interacting with glucose, and the water molecules on the other side are tied up interacting with sucrose, it doesn't matter that there's a higher concentration of sucrose relative to glucose. What matters is that the water molecules are equally tied up on both sides of the membrane. Right, so that's what osmotic pressure ends up being all about. And I've been talking now for the last few seconds about glucose, and I've suggested to you already that it would make cells happy to have glucose go from outside of the cell to inside of the cell. Let's talk a little bit about how it is that that sort of thing is going to happen. The simplest way to get that kind of movement would be passive transport. And that means going down a concentration gradient, where you have higher concentration outside than inside. If these purple balls are hydrophobic, you don't need anything to facilitate that. You'll simply get the higher concentration of the purple balls from outside to result in a net movement of molecules to the inside, because these purple balls will be happy hanging out in here and then eventually bop their way through to there. And so hydrophobic molecules like the ones that Charles Overton worked with in 1895, they don't need any help. But for water-soluble molecules like these orange triangles or these blue balls, to get across that lipidy part of the phospholipid bilayer, you need to have some sort of facilitation. And when we're talking about getting stuff done inside of cells, that means we need to, it's a cliche answer. And in fact, at some point, you guys are going to start asking me some questions. And you know what? I'm going to trot out my cliche answer, and I'll chuckle as I say it. But if ever a biologist needs something to be done They say, yeah, how does this happen? How does you do that? How did the cell accomplish this? Easy answer works almost all the time. People will just nod and say, oh yeah, very funny. A protein does it. How do you get that done? A protein does it. That's exactly what I need to trot out for you. This is perhaps the first time that you will have heard that. It's not going to be the last time you hear. How do you get these hydrophilic molecules from one side of the membrane to the other? There's a little dum-dum. A protein does it. All right? It's going to be a tired old saying before you guys are done with me in this class. But here, check it out. A protein helps get these orange triangles, where they're at high concentration outside the cell, toward where they're at low concentration inside the cell. Molecularly, it shouldn't be that hard to picture what's going on here. You got an integral membrane protein, right, hanging out, doing its thing in the membrane. Got some hydrophilic stuff up here. You got some hydrophilic stuff down here. You got some hydrophobic stuff over here. That's just the way integral membrane proteins are. But this integral membrane protein's a little bit special. It's got a hollow center. You could sort of think of it as a straw poking through that plasma membrane. And what sort of amino acids do you think might be lining the inside of the straw? They don't have to be hydrophobic ones. They could be hydrophilic ones. They could be ones that whatever this orange triangle has on its surface really likes to hang out with. Let's say this has a couple positive charges on it. You might have a couple of things with negative charges here just to make it comfortable for that to move through. This protein, this membrane-bound protein, could be something that allows just a specific kind of molecule or a subset of molecules to move through. Or it could be something that lets just anything of a certain size move through, certain size or smaller, move through. Different kinds of membrane-bound proteins can wear different hats. Some of them are very selectively permeable. Some of them are just allowing a channel for which molecules to move from high concentration to low concentration. But because a protein is involved, to help this along, you can see why this is now called facilitated diffusion. These proteins are helping, they're facilitating molecules to get from high concentration on one side across to the other side of the membrane. Okay? That works great if you're going down a diffusion gradient. The protein doesn't care. These orange triangles can move through this direction or this direction. It's just that there's more of them fighting to get through this way than this way, so there's a net movement from high concentration to low concentration. But sometimes that's not the way the world is set up. Sometimes the stuff that's on high concentration on the inside is what you want more of. You want to bring things against a concentration gradient. Glucose. Cells like to have a lot of glucose inside their plasma membranes. They often have much more glucose, a higher concentration of glucose inside their plasma membranes than what's going on outside their plasma membranes. Diffusion won't work for the cell in that circumstance. Diffusion always goes from high concentration to low. That's the opposite of what a cell would want to have happen. So how does it scarf up those last couple glucose molecules floating around outside when it's already got a lot of glucose molecules on the inside? Well, the cliche answer is a protein does it. But now it can't be just a protein does it. To go against that concentration gradient, you also have to expend energy. So a protein does it, but it does it while burning ATPs. And now we have not just facilitated transport passively, we are actively recruiting molecules from one side and bringing them to the other side, and you do that by expending energy. So that's the difference between active transport and passive transport. Passive transport, you're just riding the concentration gradient. active transport, you're using energy to physically latch onto things and bring them from low concentration to high concentration. We'll talk more about the energy and the dynamics associated with that in about a week. So, channel protein, it's a straw. Carrier protein, probably, well, it's a passive transport kind of job. Active transport, again, you're using energy to make things move from one side to the other. Here's another example of active transport. Notice there's ATP that's being used. Whenever you see ATP being used and molecules moving from one side of a membrane to another, you're talking about active transport. This is a sodium-potassium pump. If you're curious about those sorts of things, that might be something you might want to learn more about later. Here's another kind of pump that we'll talk about quite a bit in 2 weeks for this class when we're talking about how ATPs themselves get synthesized. There's actually a proton pump that's involved, where you take protons from low concentration in the cytoplasm and you pump them to higher concentration outside the cytoplasm. The trick then becomes, this is the other half of the story, we'll get into much more detail later. The trick then becomes is you have other places on the cell's membrane that allows those protons to go back by diffusion down that concentration gradient, but there you hook it up to literally, it looks at a molecular level like a little crank that creates ATPs. Right? So by pumping protons across the membrane and then allowing them to come back against that gradient later on, you can get a net gain of ATPs. Again, we'll talk about that more a good deal later. All right, so that's how you get molecules across membranes. How do you get big things across membranes? Well, you can do it with phagocytosis and endocytosis. You can just envelop them with membranes and pull them across, and everything's good. Or dump them out. Okay, I promised you I'd finish early today. I spent too much time talking about perpetual motion machines, but I'm still early, right? I've got 6 or so minutes left for the class. So let's just hit the highlights in 6 minutes or less of what I've talked with you about the last 6 weeks. Okay, let me catch my breath, and we'll go. All right, the very first time we met, I challenged you to think about what it is that we look for to make something alive. How do we know something's alive? Biology is the study of life. I suggested to you that there were at least 4 criteria that we often found ourselves looking for, but that no one of them really got the job done, and not even necessarily in combination. What were they? More organized in your surroundings, respond to stimuli, repair damage, and reproduce. Something along those lines. All right? That was pretty much the highlight, I would say, of the first time that we met. Then I started talking with you about subatomic particles. We talked about protons and neutrons. I told you that the number of electrons an atom has is governed by the number of protons in its nucleus, that the number of protons in an atom's nucleus tells you what kind of element you're talking about. If you have 2 protons, you're talking about helium. If you have 1 proton, you're talking about hydrogen. And that's the end of the story. That's the fundamental difference between different elements, is the number of protons in their nucleus. The way that elements, the way that atoms behave, is ultimately dictated by the number of unpaired electrons in its outermost shell of electrons. Hydrogen has 1, has a valence of 1. Oxygen has 2 unpaired electrons. Nitrogen 3, carbon 4. Look at that, 4 different colors. You've got a palette that you can use now to paint molecules that behave in certain ways, right? Carbon is what we turn to if we need a building block, a Lego piece that can stick to 4 other Lego pieces. Hydrogen is what we turn to if we just need something to stick to 1 other thing. Hydrogen makes 1 covalent bond, oxygen 2, nitrogen 3, carbon 4. Hydrogen and carbon, curiously, maybe miraculously, depending on your mindset, have the same affinity for electrons. And that ends up making a huge difference. It's a very important thing for the perspective of living things. Hydrogen and carbon, because they have the same affinity for electrons, their outermost shell both their outermost shells are half empty / half full. Because they have that same affinity, they share electrons between themselves equally when they make covalent bonds with each other. Equal sharing of electrons translates pretty directly to high energy bonds. Oxygen has a tendency to suck electrons from pretty much anything else that it's interacting with, at least amongst these 4 guys or 3 other guys. And that means that when oxygen is involved in a covalent bond, it's a polar covalent bond. The polarity translates to lower energy, and it also translates to a separation of charge, a little bit of negativity where the oxygen is, a little bit of positivity to whatever it is the oxygen is bound to. That partial separation of charge makes these molecules that have that water soluble. So water is a nice example of a polar covalent bond molecule. You've got an oxygen with 2 hydrogens. The hydrogens have a little bit of positive charge. The oxygen has a little bit of negative charge. It can interact very nicely with other molecules that are similar, partial separation of charge. Hence, water is a good solvent. I talked with you about, I don't know how that got there. This is just out of sequence. Forget about that. Here you go. Yeah, seriously, that was just like, whoa, where did that come from? But here, I'm showing you how it is one water molecule can interact with another water molecule. Remember, we're talking about these polar covalent bonds. The hydrogen has a little bit of positive charge. Partial positive charge is what that lowercase Greek letter delta means. The oxygen has a little bit of negative charge. The positive charge of one water molecule can rub up against the negative charge of another water molecule. Each water molecule can engage in 4 hydrogen bonds with other water molecules. We talked about how it is that once in a long while, once in 100 million or 1 billion sort of times, water molecules actually fall apart. The sharing that goes on between the oxygen and the hydrogen in the bond isn't really sharing anymore. The hydrogen just wanders off. Its proton wanders off. The oxygen keeps the electron. You get a hydroxide ion with its negative charge and a hydronium ion or a proton with its positive charge. And that in turn inspires this idea of pH. When you're thinking about pH, remember, it's a logarithmic scale, just like the Richter scale. We don't have a lot of earthquakes here, but I think you can appreciate that an earthquake that's of magnitude 6 is 10 times stronger, more shaking going on than an earthquake of magnitude 5, right? A pH of 3 means you're talking about something with 10 times more hydrogen ion concentration than something with a pH of 4. P is mathematical shorthand for the negative log base 10 of something. And the H is supposed to remind you you're talking about the concentration of hydrogen ions. So pH 4 actually means, in longer expanded form, the hydrogen ion concentration is 1 × 10⁻⁴ molar. If you'd rather, 1 × 10⁻⁴ gram per liter. Okay? And we spent some time talking about some functional groups associated with carbon molecules. There are the hydroxyl groups, the carbonyl, the carboxyl, amino, sulfhydryl, phosphate, and methyl. If you are comfortable with what I've said about polar and nonpolar covalent bonds and those sorts of valences and electronegativity, I think all of these functional groups of carbon, what you really need to know about them just sort of flows naturally from that. There is a little bit of memorization to do. The molecules that have amino groups are called amines. Ones with sulfhydryl groups are called thiols. But that's just the way it goes. Living things use 4 kinds of polymers. All right, we talk about carbohydrates, we talk about lipids, we talk about proteins and nucleic acids. Polymers are strings, long strings of building blocks that get linked together through dehydration reactions, and they get broken apart by hydrolysis reactions. The building block for an amino acid looks like this. There are 4 different kinds of amino acids: those with hydrophobic R groups, those with hydrophilic R groups, those with negatively charged, and those with positively charged R groups. The hydrophobic ones have nothing but carbon and hydrogen in them for the most part. The hydrophilic ones have hydroxyl groups or sulfhydryl groups on them. The positively charged ones have amine groups. The negatively charged ones have carboxyl groups. The building block for nucleic acids are nitrogenous bases. There's a 5-carbon sugar to which a phosphate group is attached, as well as a nitrogenous base. 2 flavors of nitrogenous bases: the ones with 1 ring that are pyrimidines, the ones with 2 rings that are purines. And we talked a fair bit about what's going on inside of cells. Prokaryotic cells are bags of goo. Eukaryotic cells are complicated bags inside of bags, and that's where we're at. All right, so good luck on Thursday. Let me know if you have some questions.